Hi, I am Yogesh Guthe. Today's technical point is drug discovery and development class. Now, drug development covers all the activities undertaken to transform the compound obtained during drug discovery into a product that is approved from launch into the market by regulatory agencies. This is a uh, pivotal process and a lot reads its success. Thus, efficiency is absolutely critical, but mainly for two key points. Firstly, development is expensive, accounting for 70% of the total R&D cost. Even throughout the number of projects is much smaller compared with those under discovery. The cost per project is significant and increased exponentially as the project progresses throughout into the later stages. Secondly, speech is the essence in the drug development as it determines how soon the company can start earning a return on the investment plugged into the project. Besides, any delay it away into the exclusivity arrangements granted by patent and time before generic manufacturers can launch Me Too products. For clarity, drug development is presented as an operation takes distinct from discovery. In reality, the distinction is not as a clear cut. Obtain different activities are being undertaken concurrently and in deep depth. Some processes that were traditional undertaken much later on are increasingly being brought much forward. The idea is to identify compounds that have the highest chances of success much earlier and focus on those. The drug discovery process technically starts with choice of a disease area and a definition of the therapeutic need that should be addressed. When this is not done, the process proceeds to identification of the physiological mechanisms that need to be targeted and ideally identification of a specific molecular drug target. During this phase, effort is focused on identifying a lead chemical structures, designing, testing, and fine tuning it and ensure that it meets all the criteria required for development into a drug product. Discovery can at first appear like a shot in a dark at the start. Scientists will be dealing with a high number of compounds which have to be filtered mainly via computer stimulations in silico into manageable numbers capable for begin further optimized. High throughput screening is then applied to identify high throughput screening which demonstrate interesting activity since high throughput screening can throw up a high number of high throughput screening. These are further optimized and validated to remove any artifacts or noise from the screen. A key aspect of validation stage is to find relationship between chemical structure and biological activity and to find out if the compound belong to an existing families of compounds known as His series. Validated hits are therefore further studied, especially in terms of their pharmacokinetic profile and toxicity. At this point, the number of compounds has reduced to a handful. It is this handful of substances that are subsequently entered into the lead optimization program. Now, lead optimization is a critical process in drug discovery since it determines whether a suitable compound can be identified for taking forward into preclinical and clinical studies. Therefore, the goal of this stage is to scrutinize and fine-tune, typically in parallel, both the biological activity and the physiochemical properties of the lead series. During this stage, rigorous data is generated in a precise, timely manner to quickly determine the compounds to progress the compounds and the series toward the ideal candidate profile. The higher the quality of these candidates, the higher the chances of successful progression into clinical trial. Thank you.